Good afternoon, sir.
Good afternoon. Just give me a minute. Let me cast my screen before we start. Is the screen visible to all of you?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, great. Amazing. So, first of all, let me tell you that when we are going to study enzymology, there are different types of concepts that we are going to come across. We will be starting with the basics of what enzymes are, how do you classify enzymes, a little bit of history of enzymes, and then gradually we will try to understand the mechanisms, the models of enzyme and substrate interaction, the kinetics part, which is a little bit where derivations and calculations come into the picture. Also, the basic mechanism of catalysis. So, all that we will be focusing on once we work on our fundamentals, because having a thorough fundamental is essential to go ahead with understanding the rest of the concepts that are there related to enzymes. So today's our session will be dedicated to work on those basics of enzymes. So we know that enzymes have extraordinary catalytic power. Okay, and this is basically much greater than the other kind of catalysts that are normally seen in case of metabolic processes. Because when you talk about catalyst, first of all, please understand that catalyst, this term stands for something which can speed up the rate of a particular process, right? So catalysts are of two types. You have organic catalyst and you have inorganic catalyst. Inorganic catalysts basically are ions, okay, which can act as agents to speed up a particular process. But let me tell you that inorganic catalysts have their own set of limitations, though they have some very very beautiful advantages associated to them, that is temperature, pH. These parameters doesn't affect them like it does in case of enzymes, but then they have their own limitations in terms of the different kinds of processes that they can speed up. But that's not the case with enzymes. Enzymes have extraordinary catalytic power. You talk about any metabolic process, you are surely going to see that an enzyme is associated with each of the reaction that you will notice in case of metabolic reactions, because every single metabolic reaction in any organism is catalyzed, which means that there is an enzyme which is working over there to carry out that reaction. So please understand that we living organisms are much lucky that our metabolic process happens at a very fast rate. And the reason behind that is these set of chemicals which we call as enzymes. Now, enzymes are very highly specific, okay? They are not going to interact with anything and everything. They have a specificity. They are going to interact with only a specific substrate. Okay, so that is something which is an added advantage which you cannot see in case of inorganic catalysts, because inorganic catalysts, they do not have such a high degree of specificity. You will see the inorganic catalyst interacting with a variety of substrates, but that's not the case with enzymes. Okay, now enzymes, they speed up chemical reactions and they function in aqueous solutions. Please understand that any enzyme, if it wants to catalyze a particular reaction, aqueous solution is what is preferable. Okay, it has to carry it out in an aqueous solution only. So even if the substrate that it might be interacting with, it may be hydrophobic, like in case of lipases. Lipases are those enzymes which interact with lipids are highly hydrophobic, but lipases still will be able to catalyze the reaction only in presence of water. And that's the reason why when you want lipases to act on lipids and hydrolyze lipids, you need to make a miscible emulsion of the lipid, wherein you add lipid and water together, you mix it well such that it forms an emulsion, because obviously the lipid will not dissolve in water, but you can emulsify it, and then you use lipases to break down the lipids. So enzymes can work effectively only in aqueous solutions, and they have some optimum requirements. Until and unless you provide them those optimum requirements, the enzymes will not function. Like, they need a specific temperature, they need a specific pH. So until and unless you provide them these conditions, enzymes are not going to efficiently function. So, these are all some of the reasons why you would see that most of the metabolic reactions have enzymes acting as catalysts, because there is a high level of specificity. They tremendously speed up the rate of the reaction. Okay, you cannot even think of the speed with which the reaction proceeds in presence of an enzyme. Please keep this in mind, that enzyme is a catalyst, it's not an initiator. It doesn't start a chemical reaction, which means that a chemical reaction can occur, it can happen even in absence of an enzyme. Enzyme is in no way an initiator. Okay, it is just going to speed up the rate with which that reaction reaches to its conclusion. Okay, so enzymes will not begin a reaction. They are going to just increase the rate with which that reaction takes place. Now, let us take a simple example. There's an enzyme called as carbonic anhydrase, found in our body, especially in the blood. Carbonic anhydrase is responsible for combining carbon dioxide and water to form carbonic acid. Okay, that's the role of this enzyme, carbonic anhydrase, when it combines carbon dioxide and water to give you carbonic acid. Now, this reaction of carbon dioxide and water combining together can happen in absence of carbonic anhydrase also. But it's going to form, like, say, 200 molecules of carbonic acid in 60 minutes. 60 मिनट यानी 1 घंटे में करीब 200 मॉलिक्यूल कार्बोनिक एसिड के आपको बन के मिलेंगे इन एब्सेंस ऑफ द एंजाइम. बट अगर सेम रिएक्शन कार्बोनिक एनहाइड्रेस के प्रेजेंस में कैरी आउट हुआ, then you will see that there are around 2 million molecules of carbonic acid that are generated per second. So now you can see the difference. 200 molecules in 60 seconds, 2 million molecules per second. That tells you the phenomenal difference in the rate of reaction that you see in presence of an enzyme. Okay, so that is the power of enzyme as a catalyst. So that makes enzyme very significant for all metabolic activities that takes place inside any living organism. Okay, enzymes work in a specific sequence. They catalyze hundreds of stepwise reactions that are taking place inside a living organism, wherein molecules are being broken down, or they are being conserved, or they are being transformed into chemical energy, or you are making some macromolecule, that is, you are carrying out biosynthesis or anabolism. You name the reaction and there's going to be an enzyme which is working there. Even though it might be the chindiest of the reactions, which you would think that, "Arey, ispe kyun enzyme kharch karna?" But even for that reaction, there is going to be an enzyme which is present. Because only then the entire rate of metabolism can be maintained at a faster pace. And that's why every single metabolic reaction you talk about has an enzyme associated to it. Please understand that these enzymes can also be regulated. And some enzymes themselves act as regulatory enzymes, which are responsible for controlling the way a metabolic pathway proceeds. It will be responsible for handling that, if a particular reaction is being carried out in excess or it is being repressed. Enzymes can do all of this. So enzymes are not just responsible for speeding up the rate of the reaction, but it could also regulate the metabolic activities that are taking place inside a cell. Now, if you ask what is the practical relevance of studying enzymes, so we have already had a brief discussion about this in the last session, wherein we said that there might be several inheritable genetic disorders where there is a deficiency or there's a total enzyme, total absence of one or more enzyme. Okay? Without enzyme, some of the reactions may not take place and they may give rise to some metabolic disorders. Okay, so in those circumstances, studying enzymes, studying their functions, understanding their significance helps us to understand these metabolic disorders even more better. Let me tell you that there are individuals whom you call as lactose intolerant. Lactose intolerant individuals cannot have products which are related to milk. So milk and milk-based food products is something which is strictly prohibited for them, because their body is not able to synthesize active enough amount of lactase enzyme, which is responsible for breaking down lactose, which is a disaccharide, a sugar which is found normally in milk. So because they are not able to break down lactose, lactose starts accumulating in their system to toxic levels. So that's the reason why they start showing you allergic symptoms whenever they consume milk and milk-related products, because they're not able to digest lactose. So under such circumstances, should we tell them to stop consuming lactose or lactose-based food products? No. You can still tell them to continue it, but you need to supplement them externally with lactase enzyme because their system is not able to produce it. So they are prescribed tablets which contain nothing but powdered form of lactase enzyme. So one tablet in a day means that they will be able to efficiently break down whatever lactose they consume during that particular day. Next day again morning, preferably consume a lactase tablet so that again for the day you will be good enough to consume lactose-based products. Similarly, there is a condition called as pancreatic exocrine insufficiency. A very complicated name, but let me tell you what happens in this particular scenario. This particular situation happens in individuals whose pancreas are not very active. So under such circumstances, they will not be able to synthesize digestive enzymes like amylases, proteases, lipases. Their pancreas is not able to synthesize any of these. So what is going to happen? Will they be able to digest food? No, they will not be able to digest food. So even if they are consuming food, it's not being digested. So the nutrients are not being absorbed. So there is going to be malnutrition that is going to take place in them because their body is not receiving nutrients even though they are consuming their food on time. So under such circumstances, if their pancreas are not producing the enzyme, it's going to be our duty to externally supplement these enzymes to them. So these individuals are going to be kept on tablets, which are amalgamation of these three enzymes, amylases, proteases, and lipases, which will help them digest whatever food they are basically consuming. So you understand that for therapeutic purposes also, a lot of enzymes could be utilized, right? In some cases, excessive activity of an enzyme could be the cause for diseased condition. Okay, and that could be really very disastrous. Like for example, if there is an enzyme which is responsible for controlling blood clotting process, and its excessive activity is leading to unnecessary formation of blood clots in the system. Now, unnecessary blood clot formation could block the arteries. If the arteries are blocked, there is going to be increase in blood pressure. Increase in blood pressure means increase in pressure on heart, and that means cardiac myopathies, heart related ailments. So एंजाइम के ना होने से जितना तकलीफ होता है, कभी के बार एंजाइम के एक्सेसिवली एक्टिव होने की वजह से भी तकलीफ हो सकता है. कई बार हम ब्लड प्लाज्मा के, इरेथ्रोसाइट्स के, टिश्यू सैंपल्स के एंजाइम का एक्टिविटी मेजर करते हैं इन डायग्नोजिंग सर्टेन इलनेसेस. So enzymes there become diagnostic parameters to check if everything is functioning properly or not. Is there any kind of malfunctioning that is taking place? Right? Many drugs exert their biological effects through interactions with enzymes. What does this mean? Please understand that काफी सारे ऐसे drugs होते हैं, दवाइयां होती है जो हम consume करते हैं in an inactive form. मतलब that form of drug cannot bring about a therapeutic action. For it to become therapeutically active, it needs to interact with a specific digestive enzyme in your body. So once the digestive enzyme acts on that particular drug, then that drug gets converted into an active form. Till then, it's an inactive form of the drug. So even that's possible. So many of the drugs which are sold in the market are basically inactive forms of drug. They become activated when you consume them. And not only that, enzymes are very important even in industries. Chemical industries, food processing industries, baking industries, agriculture, you name the area, you will find some or the other application of enzymes. So enzymes have a widespread applicability. And that makes this area extremely important to be analyzed. So let us see what's the history of enzyme research. So biological catalysis was first described in late 1700s when digestion of meat was being studied, where secretion of stomach, that is gastric juices and meat, were made to interact with each other. So that time they could see that probably gastric juice has something which has the ability to break down the components of meat. Today we know that the gastric juices have proteases which can act on protein components of the meat. So the research continued in 1800s where examinations of conversion of starch to sugar by saliva and various plant extracts was noted. Today we know that saliva has amylase, which has the ability to act on starch and break it down to sugars. And similarly, plant extracts, plants also have enzymes which have the ability to break down polysaccharides to the level of monosaccharides, right? In 1850s, Louis Pasteur concluded that fermentation of sugar into alcohol by yeast is catalyzed by ferments. So they then didn't know that what he was studying was just an enzyme decoction of yeast, which was helping to convert the sugar to the level of ethanol. Today, we refer to that process as fermentation, a part of anaerobic respiration. So he postulated that living yeast cells are producing some chemicals. He called those chemicals as ferments. And he said that these chemicals are present inside the living yeast cells. They are inseparable from the living yeast cells. So he called it as vitalism. So for several decades, it was referred to as vitalism, that the ferments, the chemicals which are there inside the living yeast cells, they act as catalysts for chemical conversions. Then in 1897, Edward Buckner was the first person to have discovered that yeast extract could ferment sugar to alcohol, okay? And that fermentation can be promoted by molecules, by chemicals, which was referred to as ferments by Louis Pasteur, even when these chemicals are separated out of the cell. Because Louis Pasteur का ये कहना था कि ये chemicals cell से अलग नहीं किए जा सकते हैं. Enzymes cell का हिस्सा है, उनको आप cell से अलग नहीं कर सकते हो. But Edward Buckner was the person to have said that in chemicals को आप cell से बाहर निकालोगे तो भी ये chemicals fermentation का काम करते हैं, तो भी ये sugar को alcohol में convert कर सकते हैं. So that was a breakthrough. Because Louis Pasteur had said that you cannot segregate these chemicals out of the cells. Well, Edward Buckner had proved that even if you separate them from the living cells, they will still be able to carry out the reaction. Frederick Kohne was the person who called these molecules as enzymes. The term enzyme came into the picture because N stands for inside and zyma stands for yeast. So the first time these chemicals were found inside the yeast, because of which the term was given as enzyma, and today we refer to it as enzymes. Okay, so with this, more and more new enzymes were isolated, their properties were studied, and that's how the science of biochemistry grew further. James Sumner again came up with a breakthrough in the early enzyme studies, wherein he isolated urease enzyme and crystallized it. And he found that even these crystals are effectively functioning. They're still showing their activity after crystallization. And he was the person to have found out that this crystal entirely was of protein as a chemical. So enzymes were chemically protein in nature. This was found by Sumner. And he postulated what is known today to us, that all enzymes are proteins, but all proteins are not enzymes. So all enzymes that we know of are proteinic in nature, except for ribozymes, where we know that ribonucleic acids, they have catalytic activity, and such RNA molecules are what we call as ribozymes, wherein they are not proteinic, they are nucleic acid, but they still have a catalytic ability associated to them. So in 1930s, Sumner's conclusion was widely accepted after John Northrop and Moses Konitz. These two crystallized pepsin, trypsin, and other digestive enzymes and found them also to be proteins. So by then people thought that maybe just urease is a protein, but how can we say that all enzymes are proteinic in nature? But as more and more enzymes were isolated and crystallized, it was found that all enzymes are nothing but proteins. So J.B.S. Haldane, this is the same person of Oparin and Haldane theory of organic evolution, who wrote a treatise entitled Enzymes. So in this, Haldane said that there is an interaction between enzyme and substrate. This interaction is crucial for the substrate to be converted into the product. He said this interaction is not very strong. This is a weak bonding interaction, but still this interaction has to be there for the enzyme to convert a substrate into a product. That was the beginning description that was put up about how an enzyme functions. But unfortunately, at that point of time, it was not very well accepted. But today we know that enzyme and substrate have to interact with each other. They have to form a complex. Only then the enzyme will be able to show its catalytic activity, convert the substrate further into a product. Since the later part of 20th century, the research on enzymes has been extensively vast, because you see every single lab, they are working with some or the other enzyme. So there is so much of work that has happened in the past decade, or past, you can even say century, regarding enzymes, that the enormous amount of data that we have regarding enzymes comes from all those research studies which were carried out independently in research laboratories. Now we have come across to such high-end technology that we are able to purify a variety of enzymes, we are able to elucidate its structure, we are able to find out the chemical mechanism by which it functions, and we have, from all this knowledge, we have found out an empirical understanding, a general mechanism by which the enzymes function. So thanks to all those researchers out in the community who have invested their time and money in understanding the basic structure, the basic functioning of enzymes. Otherwise, we would have not had the preliminary data also that we have today. So with exception of a small group of catalytic RNA molecules, as I said earlier, all enzymes are proteinic in nature. Their catalytic activity is going to depend on their structure. We always say this, that the structure of the enzyme is extremely important, because the structure has to be definite for the function to be definite. If the structure is getting affected, the function of the enzyme is also going to get affected. So the proteinic configuration, the native structure of the enzyme is very, very important to understand its activity. If an enzyme is denatured, if an enzyme is dissociated into its subunits, then you cannot expect the enzyme to show its activity. So if the structure is lost, the catalytic activity is also going to be lost. If an enzyme is broken down into its component amino acids, then its catalytic activity is totally destroyed. कई बार ऐसा होता है कि एंजाइम मल्टीपल सब यूनिट से बना होता है, एक पेप्टाइड से नहीं 4 पेप्टाइड से बना हो, 2 पेप्टाइड से बना हो. तो अगर आप वो 2 पेप्टाइड को अलग कर देते हो एक दूसरे से, तो ऑब्वियस है कि एंजाइम का एक्टिविटी चला जाएगा. लेकिन शायद अगर आप उन दोनों पेप्टाइड को साथ में दोबारा लेकर आओ, तो शायद एंजाइम काम करने लगेगा. Maybe activity may not be as efficient as it was earlier, but there is going to be some amount of activity that is restored if you join those peptides back again. But if you're going to break down the peptide to the level of single amino acids, oh, then it's gone. Then you cannot even expect the catalytic activity to return. The catalytic activity will be lost. Okay, so the primary, secondary, tertiary, and quaternary structure of the protein enzymes is very, very essential for the catalytic activity of the enzyme. So, like other proteins, you will see that enzymes have high molecular weight. They could range from 12,000 to more than 1 million, depending on the enzyme that you're working with. Some enzymes, they do not require any chemical groups other than their amino acids. Just the proteinic structure is more than enough and the enzyme is active. But for some enzymes, other than their proteinic component, they may require an additional chemical component. And that's what we call as cofactors. I'm sure you've heard of this term. Cofactors basically are one or more inorganic ions like ferric ions, magnesium ions, manganese ions, calcium ions, zinc ion, cobalt ion. So an enzyme will have a definite cofactor, one or more, but definite. It's not that magnesium nahi hai to chalo calcium se kaam chala lete. ऐसा नहीं होता. Magnesium चाहिए तो magnesium ही चाहिए. Without that cofactor, the enzyme is not going to show its activity. Okay? Sometimes, along with the cofactor, the enzyme may need another chemical component. And that chemical component is not inorganic, it's not ionic. It's an organic or a metallo-organic molecule, which we call as coenzyme, like vitamins, reducing agents like NAD, FAD. This you would have all heard in biochemistry from whichever biological sciences you come from. NAD, FAD, you know that these are agents which accept hydrogen and form NADH and FADH2. Very important in the process of respiration. So all these vitamin-containing components can act as coenzymes. So either the enzyme could require cofactor or it could require coenzyme or a combination of both. And mind you, if the enzyme doesn't get the cofactor or coenzyme that it needs to function, the enzyme will not show its catalytic activity. It is a must. So if it needs niacin as a vitamin, it needs niacin. You cannot say use cyanocobalamin. It will not do that. Because please understand that enzymes have specific sites on themselves where these cofactors and coenzymes can come and bind. And those sites are shape specific. So if that site is according to the shape of magnesium ions, you cannot fit manganese ions over there. It's incompatible. So magnesium की जगह कोई और नहीं ले सकता. वहाँ पे magnesium ही चाहिए. Same stands for coenzymes as well. Is all this making sense to you? Can I see a yes or no in the chat box, please?
Yes, sir.
Others, out of 22 people, I get 2 answers. Not
yes, sir.
Great, amazing. Okay. So this I can relate to you guys from the point of view of a physiological scenario. Many a times this happens that you feel physically tired and exhausted. And you see that you have not been doing some heavy physical activity to feel so. Okay? So that tiredness seems to be a little abnormal. You don't know why you are feeling that way. So you decide to go to the physician, you go to the doctor, the, the doctor hears you out, and then what he does is he just plainly writes down vitamins and mineral tablets for you and says that please consume this for the next 15 days. And once you consume those, you feel good enough. And you're like, wow. But you don't think that what did they actually do? Please understand that your reason of fatigue in this case could have been deficiency of specific vitamin or minerals, which means deficiency of cofactors and coenzymes, which means that your respiratory enzymes, which require cofactors and coenzymes, probably were not functioning with the capacity with which they can normally function. So the amount of ATP synthesized in your system is much lower than what it can be. So once you start consuming those vitamin and mineral tablets, what are you doing? You are replenishing for those vitamins and minerals which are deficient in your system. And that makes you feel energetic again because now the ATP reserves are restored. So see, the basics that you learn over here, you can extrapolate it to your day-to-day life. And that's when, you start understanding concepts, you start clicking with the concepts, and then it remains with you for the rest of your life. You'll never forget it because you have now started relating the thing to a particular example. So do that. That will help you in the long run to, recollect concepts, reiterate it, and mention it out. Okay, just give me 2 minutes. I'll be back. Just give me 2 minutes. Okay, so as said earlier, you can see that some enzymes require both a coenzyme as well as cofactor and maybe one or more metal ions also for its activity to be seen. Okay, now a coenzyme or a cofactor is very tightly, at times covalently linked to the proteinic part of the enzyme. And this coenzyme or cofactor which is attached to the proteinic group of the enzyme is what we call as a prosthetic group. So cofactor or coenzyme comes under the category of a prosthetic group. Okay, so a complete enzyme is protein plus its cofactor or coenzyme. That is what we call a complete enzyme. The complete active enzyme is the proteinic part plus its cofactor or coenzyme. And this complete active enzyme is what we call as hollow enzyme. Okay, it's what we call as hollow enzyme. The proteinic part of the enzyme is what you call as apoenzyme or apoprotein. The cofactor or coenzyme is called as prosthetic group. So I can say that hollow enzyme plus apoenzyme, or rather, apoenzyme plus the prosthetic group is going to give me the hollow enzyme. See, what is hollow enzyme? A hollow enzyme is a completely active enzyme which consists of the proteinic part, the cofactor, the coenzyme. Just the protein part of the enzyme is what we call as apoenzyme. Just the cofactor or the coenzyme is what we call as prosthetic group. So apoenzyme plus the prosthetic group, which means the proteinic part plus the cofactor or the coenzyme, is what makes the complete enzyme, which we call as the hollow enzyme, the whole enzyme. Okay, coenzymes act as transient carriers of some specific functional groups. Most of the coenzymes are derived from vitamins or some or the other organic nutrients. So this has to come from diet. So if your diet is not balanced, if your diet doesn't have enough vitamins, there's going to be a subsequent deficiency of coenzymes in your system. Also understand that at times enzymes may be modified by adding a phosphate group to it, which you call as phosphorylation, or by adding a sugar group to it, you call it as glycosylation, or some other kind of covalent modification is possible. Many of these alterations may also be responsible for regulating the activity of the enzyme. So whenever you covalently modify it, by adding a phosphate group, or at times by adding a methyl group, or by adding a sugar residue, then it could also be in a way regulating the activity of the enzyme, either inactivating it or activating it. Okay, so it's like a switch off, switch on mechanism. Enzymes are always kept under tight regulation. Remember this, because if the regulation over the enzyme is lost, फिर सत्यानाश है, सब बर्बाद हो जाएगा. So there has to be a tight regulation over the enzyme. And there are mechanisms in place for regulating the enzyme activity also. Many of you would have learned as a part of biochemistry in your respective undergraduate courses as well. Right? Now this table tells you, for in cases of some of the enzymes, which are the cofactors. So you can see that for cytochrome oxidase, you have copper ions. Cytochrome oxidase, catalase, peroxidase, you can have iron Fe2 plus or Fe3 plus. Pyruvate kinase, you have potassium. Hexokinase, glucose 6-phosphatase. Pyruvate kinase, you have Mg2 plus. Aragonase, ribonucleotide reductase, you have Mn2 plus. Dinitrogenase, which is very important enzyme for nitrogen fixation process, you have molybdenum. For urease, you have nickel ions. For glutathione peroxidase, you have selenium. And for carbonic anhydrase, alcohol dehydrogenase, carboxypeptidase A and B, you have zinc. So you can see that for different enzyme, different cofactors are needed. You're not expected to by heart this. There's no need to by heart which cofactor is working with which enzyme. हम यहाँ रट्टा मारने के लिए नहीं आए. The purpose of this table is to make you realize that the cofactor is not the same for all enzymes. You can see that with the different enzyme names, the cofactors also vary. You can also see here they have basically told you coenzymes, and they have told you what chemical group, what functional group they carry, and which dietary component provides it. All coenzymes there have been mentioned. So which vitamin or which organic compound gives rise to that particular coenzyme has also been mentioned there. And that coenzyme is responsible for carrying what? That has been mentioned over there. So you can see biocytin comes from biotin, which is a vitamin again, carries carbon dioxide. Coenzyme A, acetyl coenzyme A, if you remember from Krebs cycle, carries acyl groups, comes from pantothenic acid and other compounds, again a vitamin. Deoxyadenosyl cobalamin, which is also called as coenzyme B12, carries hydrogen atoms and alkyl groups, comes from vitamin B12. Flavinadenine dinucleotide carries electrons, comes from vitamin B2, which is riboflavin. Lipoate carries electrons and acyl groups, not necessarily coming from diet. Nicotinamide adenine dinucleotide, NAD, carries hydride ion, comes from nicotinic acid, niacin, vitamin again. Pyridoxal phosphate carries amino groups, comes from pyridoxine, which is vitamin B6. Tetrahydrofolate carries one carbon groups, comes from folic acid, again vitamin. And thymine pyrophosphate carries aldehydes and comes from thymine, which is vitamin B1. So enzymes, how are they classified? They are classified by the reactions that they carry out. You will always see that enzyme names, they end with the suffix ase. Lipase, protease, amylase. So always the suffix is going to have ase associated to it. Okay, so depending on which substrate it catalyzes or which process it carries out, on the basis of that, enzyme names will differ. So urease catalyzes hydrolysis of urea, hence the name urease. DNA polymerase catalyzes polymerization of nucleotides to form DNA, so DNA polymerase. Some other enzymes are named by their discoverers for a broad function. Okay, for example, an enzyme that is known to act in digestion of food was named pepsin, from the Greek word pepsis, which means digestion. But we know that pepsin actually acts only on proteins, but it has got such a broad name, right? Lysozyme was named after its ability to lyse bacterial cell walls, hence lysozyme, right? Still others, they may be named on the basis of their source, like trypsin. Okay, this comes from a Greek word tyrene, okay, which means to wear down. So basically it is obtained by rubbing the pancreatic tissue with glycerine. That's how trypsin is obtained. So you're wearing the tissue down by constantly rubbing it. That's how it got its name as trypsin. Sometimes the same enzyme may have one or multiple names associated to it. Or two different enzymes may have the same name. Even that has happened in the past. And because of such ambiguities, an ever-increasing number of enzymes being discovered every single day, there had to be a proper taxonomy system which will be universally adopted for naming and classifying the enzyme. So this system divides the enzyme into 6 classes, and each class is further divided into subclasses, and that is on the basis of the type of the reaction that it catalyzes. Okay, let us see those classes first before we discuss the rest 2 points on that slide. Oxidoreductases, as the name suggests, they are responsible for oxidation reduction reactions, redox reactions, transfer of electrons or hydride ions. Transferases, as the name suggests, involved in transfer of groups, chemical groups. Hydrolases, responsible for hydrolysis reaction. Lyases, which is responsible for breaking double bonds or formation of double bonds. So their lyases is involved. Isomerases, wherein it is responsible to carry out isomerization, converting one compound to its isomeric form. Ligases, which are responsible for bringing about condensation reactions, joining stuff, formation of bonds, new bonds, that's done by ligases. So please understand that each enzyme has been assigned a 4-part classification number and a systematic name by the International Enzyme Nomenclature Committee. And that number helps you identify what the reaction, which reaction is catalyzed by this specific enzyme. So let us take an example to understand that. So look at this reaction, ATP plus D-glucose, you get ADP plus D-glucose 6-phosphate. So the enzyme which functions here is called ATP glucose phosphotransferase. Now, we also use an enzyme named hexokinase for this. But the systematic name is ATP glucose phosphotransferase because this name indicates that a phosphoryl group was transferred from ATP to glucose. So ATP glucose phosphotransferase is the systematic name. And there is an enzyme commission number associated to it, which is called as EC number. So every enzyme has a number and a systematic name. It's a 4-digit number, 2.7.1.1. And each number here has a significance. Like the number 2 denotes the class to which this enzyme belongs, which is transferase, because see, it is carrying out transfer, right? So transferase. So that's the second class of enzymes. The number 7 denotes the subclass. What does it transfer? Phosphate group. So phosphotransferase. So amongst transferases, it belongs to the 7th subclass, which is phosphotransferase. The third number 1 indicates that it's a phosphotransferase with a hydroxyl group as an acceptor. So the phosphate group that it transfers from ATP to glucose is first accepted by the enzyme at the hydroxyl group. That's indicated by number 1. And yet another number 1 indicates that D-glucose is the acceptor of phosphate, of the phosphoryl group. So 2 indicates the class of enzyme, 7 indicates the subclass, 1 indicates that which group is acting as acceptor, and the last one indicates that which compound is accepting the phosphoryl group. So that's how the 4-digit number classification works. Have you understood this?
Yes, sir.
So now, as I said that in this case, there is a trivial name for the enzyme which is very commonly used. That's hexokinase. But that's not the systematic name. That's like a common name. जैसे अपने biological organisms के लिए होता है वैसे. तो ऐसा complete list and description of thousands of known enzymes is maintained by Nomenclature Committee of International Union of Biochemistry and Molecular Biology. And you will find that list on this particular website. So no one obviously ratifies all this. No one does that. कि कौन सा नंबर कौन से एंजाइम का है, कोई नहीं करता. वो डेटाबेस में अवेलेबल होता है. बस हमको पता होना चाहिए कि नंबरिंग सिस्टम किस बेसिस पर दिया गया. That is more important. Okay, so we will stop at this point today. If there are any questions, please let me know. And in the next lecture, we will proceed ahead with the discussion on how enzymes work and some more terminologies related to the enzyme. So if there are any questions till this point, please do let me know, or else we are good to stop.
Okay, sir. Thank you.
Anyone else? Any questions? If no, just type a no so that I know that there are no queries and we can stop.
No, sir. No questions.
Okay, great. So have a great weekend and see you all in the next week. And we will discuss more about enzymes then. Thank you so much.
Thank you, sir.
Thank you,
Thank
sir.
you.